Okay, fine. Hope everything is going well. I'm giving in the full screen mode. Can you see the slide? General virology, picture of two viruses are there. Okay, fine. So once again, I welcome all of you to this particular session. This is in continuation of our discussions on the virology, general virology. So in the previous class, we talked about some of the historical developments, some of the milestones and important events that happened in the development history of veterinary virology. And thereafter, we start giving some discussion about the viruses and how they differ from the other living organisms. As such, why we don't call it as an organism, rather than we call it as a particle, virus particle. So basically, these two kinds of viruses, the most simplest forms, the naked nucleocapsid virus, simply we call this a nucleocapsid structure of the viruses. There are many viruses which are existing in this nature simply as nucleocapsid structures. So on the left-hand side, you can see this is a nucleic acid covered with a protein coat called as nucleocapsid. However, I mentioned about some other agents called as virusoid. Virusoids are certain plant viruses where even they don't have the capsid layer, only the nucleic acid is the entity. We call them as the virusoids. Okay. In fact, we don't have any virusoids infecting animals in the human. So we call them as the virus. And all of you know, and a fully matured infectious form of the virus, we call it as a virion. Okay. So some other kind of viruses which additionally acquired a layer from the host cell during its replication, where they can embed some of their proteins like spike proteins or peplomers. We call them as the envelope viruses. So basically, the viruses belonging to non-envelope and envelope viruses. So we talk about why it cannot be seen because of it's beyond the resolving power of the light microscopes. We cannot see it on the light microscopes. We need electron microscope with high resolution and high resolving power. So the smallest is the circoviruses, around 17 nanometer in size. Then the biggest one of our important, the pox viruses, 250 to 300 nanometer in their size. Okay. So what we discussed in the previous class is that the viruses do not have their metabolic activity. That's a very interesting features, because of which we are in a dilemma whether we should call it as an organism or not. Okay. However, at the same time, one virus can replicate and give rise to an in vitro similar kind of viruses. From one virus, thousands of virus particles can be formed. That is again characteristics of living things. So we call the viruses they are at the age of living. These pictures we try to explain like the other higher the eukaryotic cells is having lot many organelles. They're having a cytoplasmic soup, and a lot of metabolic activity takes place, and independently they can grow and divide. They can take nutrient from the surroundings. Similar case with the bacteria. With limited organelles, they independently can survive and can accumulate nutrient from the surrounding. However, in case of the viruses, they don't have any cytoplasmic soup, or as such, no metabolic activity will find in a virus. Inside a virus, no organelles. It's simply a nucleic acid which is protected by a protein coat. We call it the capsid layer, and together we call it the nucleocapsid structure. It is the most simplest form of virus. In addition, the envelope viruses, they have the additional envelope and the matrix layer, etc., etc., and they can exist in nature, but they don't have any metabolic activity of their own. So that's why we call that the viruses, they are at the age of life. And these are the things that we discussed in the yesterday's class, and how, and a very interesting group of viruses called as the retroviruses, which can get integrated with the genome of the host cell. They can get integrated to our chromosome and can survive. So this features is never seen in any other living system. That's why we call the viruses, they are at the age of life. So lastly, we try to compare about the viruses and the bacteria with certain features. Hopefully all of you can understand and record these presentations. Now, it's a very small area. The origin of viruses. Obviously, people will say the evolutionary process, like from the unicellular organisms, how the plants and animals were evolved in the evolutionary process. The obvious question is that where from the viruses came to this world. So again, there are a lot of opinions and thoughts. Some school of thoughts has been developed who are exploring like where from the viruses has come. You'll be amazed to know that in Spanish flu that occurred in 1915 to 1917, people thought that those viruses were simply soaring from the sky. It's coming from the alien world, and that's why globally the disease is occurring. But in fact, that is a wrong concept. So people were thinking like where from the viruses were coming to the nature. So in a very short way, I'll try to explain it. Some people thought that they are cellular gene robbers, means the viruses evolve from the host cell. That means from higher plants and animal, they simply steal some of the genes and they get into an identity of viruses. As we could see in the retroviruses, since they can integrate it with the host cell chromosome, obviously they can come out of the host cells. So that says that they might be the cellular gene robbers. But at the end, if you talk, then you'll find the chicken or egg dilemma, which has come first. So even the origin of you at certain point where from the viruses came to the world, then these are the three probable hypotheses. People believe that either one may be there, or even the last point said no single hypothesis may be correct. So still it is not clear like where from it has come. But looking at the nature of viruses, one of the theory called progressive hypothesis, where it is believed that some of the mobile genetic elements of our higher plants and animal somehow escaped from that cells and give rise to viruses. Some structures like retrotransposons, which are called as the mobile genetic element, they can also called as a jumping gene. Within the chromosome from one part to the other, these genes can move. And like 42% of the human genomes, you will get this kind of mobile genetic element. So this school of thought says that probably this retrotransposons or mobile genetic elements have escaped from the host and that give rise to a virus identity. And this is what we call as the progressive hypothesis. However, another school of thought says that it's not that way the virus has evolved. It is rather a regressive hypothesis, where they say that probably the life system has already evolved in this world, and then some of this gradually has losses their genetic material gradually, and they become they lost the independent replication process in the world, and finally they become attached to an host cell. They become totally dependent on their host cell. So that means regressive. They are missing some information, or genetic information in the evolutionary process, and they become from higher to the lower form of life. This they try to correlate with this two group of bacteria, Chlamydia and Rickettsia. They are bacteria, but gram-negative bacteria, but they do not have all the metabolic activities of an free-form bacteria. As a result, Chlamydia and Rickettsia, they cannot be grown in the artificial media. They always need a living cell, just like the virus, to replicate and to survive. So that's what we call as a regressive hypothesis. Probably this from certain organisms, they loses some of its essential gene for its survival in the nature, and they finally give rise to viruses. This is called the regressive hypothesis. There are a certain group of viruses, it's called nuclear cytoplasmic Lers DNA viruses, like pox virus, like herpes virus. They sometimes they're having some independent natures inside the host cells they can replicate. So they are more or less some similarity with this group of bacteria. So it's called as a regressive hypothesis. And the third school of thought say that no, it's the co-evolutions. As the life exists in the world, right from the beginning, it was the co-evolution, the viruses were there. So these are the recently they published in the Nature about this probable hypothesis where from the viruses came into this world. So this was information you can keep with you. Now straight away we'll go to the structure and morphology of viruses. As Subrisa told that he has covered most of its part, but still taking it as a revision, so we will again discuss about the structure and morphology of viruses. The first thing all of you must understand, and you note it down, virion, V-I-R-I-O-N, virion. Virion means an infectious virus particle. A complete virus particle is called as a virion, okay? So if you see the most simplest virions, it consists of a nucleic acid, either DNA and RNA, which are protected with a protein coat called as the capsids, and together we call the nucleocapsid structures. Again, I'm repeating, it is the simplest form of the virus. There are many viruses, they are existing in this world simply as nucleocapsid structures. Whether we talk about the adenoviruses, circoviruses, papoviruses, all are the simple nucleocapsid structure virus. However, there are a certain category of viruses, in addition to these nucleocapsids, they acquire an envelope layer, which straightway they steal it from the host cell membrane, and that gives rise to the envelope group of viruses. Okay. Now, if you go to the composition of viruses, straightway we can say in terms of the nucleic acid, proteins, and carbohydrate and fats, whatever is there, they make the viruses. So if you see the major part of the virus, it's the nucleic acids, and we know that the nucleic acid of viruses are either DNA or RNA, never both, okay? This point I will request you to note it down, because very frequently we ask about this characters of the virus nucleic acid. Okay, if you had any issues, please you can call me or you can text me. So the viral nucleic acids, either DNA and RNA, it may be a single molecule, we call them as the monopartite. All the genes are present in the single molecule. Or there are certain RNA viruses where the DNA or RNA is a multipartite or segmented. Of course, we don't have any segmented DNA virus, only among the RNA virus. There are certain RNA viruses where the nucleic acid is segmented. They are having certain 10, 11, 12 different segments in case of the reoviruses, biRNA2 segment, orthomyxoviruses, which include the influenza viruses of animals and humans, or any other marine animals also. We have the influenza viruses, which is having 6, 7, or 8 segments depending upon their genus, different genus. So all the genes of the organisms are distributed in these different segments. So monopartite and multipartite, these are the two words frequently we use to describe the physical nature of this viral genome. Remember, we don't have any segmented DNA viruses. Segmented or multipartite nature is present only in certain RNA viruses, not all. Then again, the point, all viral genomes are haploid, means they carry only one copy of each gene. Almost all viruses, they are having haploid, except one category of viruses. We call them as the retroviruses. Retroviruses, why they are important is that they are having the reverse transcriptase enzyme, and they can get integrated with the host cell chromosome. The processes that include the disease like bovine immunodeficiency, human immunodeficiency, then you're having disease like tumorigenic infections, avian leukosis. So all these retroviruses, they are having diploid genome, means each of the genes in duplicate copy is present. Now, this is again established the theory like how they have jumps out from the animal chromosomes. So the eukaryotic genome is having the diploid nature. So the retroviruses, their genomes are also diploid in nature. Please note it down, all these points, highlighted points. Then again, the DNA viruses, among the DNA viruses, the genome, it may be a linear molecule. Linear, both the ends are open, or circular. There are certain circular supercoiled DNA, which is the nature of plasmids or bacterial chromosome. We could see among the DNA viruses. So DNA viruses genome may be a linear or a circular molecule. So polyoma, papilloma, this viruses, tumorigenic DNA viruses, it's circular and supercoiled. Okay. Whereas the virus, DNA viruses like the circoviruses, the enteroviruses, they are also having circular genome. Okay. Whereas in case of the RNA viruses, we don't have any circular. All RNA viruses, they are linear. Both the ends are open. Okay. Now coming to one of the most interesting feature, like which you could not see in any other life system. Here you'll find that universally, all of you know DNA are the double-stranded molecules. The Watson and Crick model, everyone of us, we have studied the double helical structures of DNA. Then DNA, we know they are double-stranded and RNA are single-stranded. But there are two DNA viruses of our importance called as parvoviruses and circoviruses. Parvo, circo, enteroviruses. These are the example of DNA viruses, but they have single-stranded DNA. Single-stranded. We write it as ssDNA, single-stranded DNA. So it is like quite peculiar things. In nature, no DNA are single-stranded, but these two group of viruses, their DNA is single-stranded. Please note it down. So genome of all DNA viruses are double-stranded except parvo and circoviruses, where it is single-stranded. There is another DNA viruses. We call it a HEPA DNA virus. It's causing one major infection in human, but one member is causing infections in ducklings. So that HEPA DNA is the name derived from the main clinical manifestation, hepatitis. It causes severe hepatic damage. HEPA DNA viruses, the genome is partially double-stranded and partially single-stranded. This is quite a phenomenal things we could see in the viral DNA. Similarly, when we talk about the RNA viruses, you'll find that majority of the RNA viruses, the RNA is single-stranded, as all of you have studied that all RNA are single-stranded. Yes, in fact, majority of the RNA viruses, the genome is single-stranded. However, there are two categories of RNA viruses of our importance. One is the reovirus, and other is biRNA virus. This two group of virus possesses double-stranded RNA. Okay, so these are some of the very peculiar things that we could observe among the viruses. Now, coming to some other characteristics of viral genomes is that some of the most simplest viruses, DNA viruses like the Circo or Aniloviruses, were having the genome size of 1.7 kilobases. 1.7 kb is a very, very small. This is a highly questionable things like the entire genome, which is 1.7 kb, how an organism can survive? Can it be an organism? So these are some of the questions very difficult to answer. So this Circo viruses, hardly they are having 4 to 5 different proteins, and with that proteins they are happy and they can survive in the nature in comparisons to higher plants and animals, like it's beyond comparison. So the DNA viruses with the very simplest, simple or the very short genome length of 1.7 kb to some of the higher kinds of viruses like African swine fever virus and pox viruses, where the genome is very, very big, almost 200 kb size. They carry a lot of genetic information. However, the simplest viruses carry very limited genetic information, and with that information, they can survive in nature. Similarly, if you see the ranges in the RNA viruses, the largest almost, sorry, it's not 2.9, it's 29. Please correct it. It's not 2.9, it's 29 kb. This coronaviruses and arteriviruses, they are having the size of this 29 kilobases. Then simplest one is again 1.7. So a wide size of the genomic constitution we could see among the viruses, both DNA and RNA. Could you see the slide properly?
Yes, sir.
Okay. Anyone still waiting to join the class? No. Okay. 49. Yeah. 49 students has joined. Now coming to the point, the last point mentioned in this particular slide. The RNA virus genome is characterized among the RNA viruses as positive sense or negative sense, sometimes called as polarity, positive polarity or negative polarity. The viral genome. Now, what is this polarity or sense? This is quite important. All of you must record it properly. So there are certain RNA viruses where the viral genomic RNA itself acts as messenger RNA. Hopefully you understand. In nature, what happened? The DNA carried the genetic information. From the DNA, a messenger RNA is transcribed using the DNA-dependent RNA polymerase, and that messenger RNA carried the genetic code, and on the surface of ribosomes, the protein gets translated. That's the normal process. So you have heard that all system, the transcription is essential. But in case of positive sense RNA viruses, the viral RNA itself or the sequence of viral RNA is similar to that of the messenger RNA. So no transcriptions occur in this group of viruses. So as soon as the virus genome enter inside the host cell, protein synthesis takes place without causing any transcription. So those viruses, RNA viruses, we call them as the positive sense or positive polarity. Again, I'm repeating, positive sense, single-stranded RNA viruses are those viruses where the viral RNA is of same sense to that of messenger RNA. So once they enter inside the host cell, the protein synthesis they can initiate. Got my point? Now coming to the other categories called as the negative sense RNA viruses. So this negative sense RNA viruses, their sequence is not similar to that of the messenger RNA. So what happened? They have to undergo transcriptions. They have to synthesize messenger RNA from the genomic RNA. Here they faces major issues like we have studied, all of you studied in the nature, from an RNA, another RNA cannot be formed. RNAs are formed from the DNA molecule. So as a result, what happened? This negative sense RNA viruses must possess a very special enzyme called as RNA-dependent RNA polymerase enzyme, which is not present in the eukaryotic system. Our body cells, animal body cell doesn't have this enzyme, RNA-dependent RNA polymerase. So where from they will get this enzyme? They must carry, this group of viruses must carry this enzyme for transcription. So utilizing this enzyme, RNA-dependent RNA polymerase, the transcription occurs. From a genomic RNA, a messenger RNA is synthesized and proteins are translated. So this is quite interesting, and we call this group of viruses as negative sense RNA viruses. So the RNA genome of RNA viruses, either it will be a positive sense or negative sense. Okay. In case if it is a negative sense RNA virus, it must possess this RNA-dependent RNA polymerase enzyme for transcription. So this is what we call as the sense or polarity of viral genome. The last point, this is a unique thing that presents in the retroviruses, only in the retroviruses. The name itself indicates retro. It's the RE stands for reverse. TR comes from transcriptase. This group of viruses, retroviruses, they possess the enzyme reverse transcriptase. RE and TR. This has come and give rise to the word retro. So they possess an enzyme called as the reverse transcriptase, and more specifically, if you say, then we'll call RNA-dependent DNA polymerase enzyme. Again, these things never happen in any higher life system. Our body cells, animal body cell, never undergoes transcriptions of DNA from RNA. The reverse way never goes. But in this group of viruses, it happened. They possess this enzyme. And as soon as the retrovirus enter within the body cells, utilizing this enzyme from the RNA, they will convert it into double-stranded DNA, and then they get integrated with the host cell chromosome. So that's the very, very peculiar things that we could see among the viral DNA and RNA. Some of the interesting points. So hope that all of you have recorded very properly. So in the subsequent classes, we'll discuss more about these groups of viruses, their transcription strategies. Now coming to the proteins. So what are the different viral proteins you'll see within the virion, and virion, how it is constructed, okay? So you'll find among the viruses, the simplest form of the viruses having very few protein, as I say, 3 to 4 proteins, they compose a virus, the simplest one. However, the pox and the herpes viruses, as for viruses, African swine fever viruses, they are large DNA viruses. So they have more than 100, even 200 different proteins are present. So to understand this protein present in the viruses, it can be very easily categorized into two important groups. One is called as the structural protein and non-structural protein. All the viral proteins we can categorize and call them as structural and non-structural protein. So it's quite obvious, and everyone can get an idea. When we talk about a structural protein, that means it will help in formation of the virus structure. As we could see, the nucleocapsid structure, those capsid layer are the structural protein. Okay. Whereas in case of the envelope viruses, even they have some proteins like matrix protein. We had this picture. This is the nucleic acid schematic diagrams, and the capsids protein, they're forming the capsid layer. So these are all structural protein. Okay. In case of the envelope viruses, we could see this brown colored area. This is the envelope. This is not a viral origin. They simply acquire it from the host cell during replication. This brown layer, what we could see. However, this structures what we call as the peplomers or spike. These days, all of you have heard about the spike proteins of this coronaviruses. These are the spike proteins. These are of viral origin. This red layer is the envelope, which they simply acquire from the host cell during replication. But this envelope or some of the proteins get punched to the envelope. We call them the spike proteins or glycoproteins. So these are of viral origin. These are again structural proteins, spike proteins, and they are again formed. They adhere to one layer, what's called as the matrix protein. This mass color, you can see the layer. This is the matrix protein. So these are the example of structural protein. So what I said that all the viral proteins, we can categorize them as the structural and non-structural protein. Structural protein is quite visible, and they give rise to the structure of the virion. The other category of proteins we call as the non-structural protein. These non-structural proteins are mainly enzymes or certain factors which are needed for replications. It's needed for that proteolytic cleavage, assembly of the virus within the host cell before it out of the host cell, and polymerase enzymes for genomic replication. So even certain large DNA viruses, we are having a group of non-structural protein called as virokines, which again are type of protein that helps to establish virus infections. They act against the host innate immunity. We'll discuss this point later on. But what you should remember is that the non-structural proteins are mostly the enzymes. They are needed for genomic replication, gene regulation, proteolytic cleavage, assisting assembly, etc., etc. So these are synthesized once the virus get entered into the host cell, and they are produced, and utilizing this, the virus can complete its replication within the host cell. So all the virus proteins we can just categorize into structural and non-structural. So capsid, matrix, even the glycoproteins, these are the spikes or encoded into the envelope, called as the glycoproteins. These are all structural proteins. So this, again, the capsids, matrix, and glycoproteins may remain in modified and unmodified proteins. Okay, so some of the proteins as such, they participate in formation of the virion structures, but there are certain proteins, more particularly the glycoproteins, as we are talking about the spike proteins of coronaviruses or classical swine fever, etc. So many viruses, the glycoproteins, which are the spike proteins, are mostly helps in establishing infection in the host cell, and in fact they are again good immunogens and our immune system can recognize these glycoproteins and can elicit protective antibody. At present, the Covishield vaccines, which most of us we have taken it and available in the markets, are prepared by synthesizing these spike glycoproteins of coronaviruses. These are the spike glycoprotein. It has been seen that since they are present on the surface and they are very good immunogens, our immune system elicit antibody against these. So in this Covishield vaccines, using some adenoviruses, the recombinant adenoviruses were prepared by utilizing the genetic information of this spike glycoproteins, and these glycoproteins are synthesized by recombinant DNA technology, and that are used. This spike glycoproteins are used as a vaccine instead of the whole virus. So that becomes a very safe form of the vaccines. So these glycoproteins, they are very, very important in terms of the virus as well as in terms of our concern, okay? So viruses also essentially need this glycoprotein to establish infection inside the host. They can bind with the receptor to initiate the penetration and entry of the virus. However, at the same time, our immune system also can recognize this very properly and can elicit antibody production against it to get protection. So these glycoproteins are mostly modified proteins. Once they are synthesized inside the host cells, then they undergo some sort of modification. Particularly, they are linked up with certain carbohydrate molecule. We call it as a glycosylation process, and those modified proteins remain integrated with the membrane. So type 1 and type 2 membrane proteins are the categories of viral glycoproteins. Okay, I can see this structure. This is nucleic acid. This is the nucleic acid structure. This is an helical symmetry. We'll come to that point. However, don't get it confused with this guy. This one is adenovirus. This is a non-enveloped viruses. They're also having some sort of projections, look like spikes, but these are not the enveloped glycoprotein. These are the viral proteins found in the penton area and called as the penton fibers that present adenoviruses. As you know, this Covishield vaccine is prepared by utilizing a recombinant adenovirus where this spike glycoprotein gene has been incorporated, and these recombinant viruses are producing the SARS-CoV-2 glycoproteins in the cell culture. So that some of the times can be discussed. Now, if you see the functions of structural proteins, then you'll find basically it gives us protection to the internal structures of the virus and give it its energy so that the nucleic acid remain protected, doesn't get inactivated by the external components. Okay. Moreover, the structural proteins also act as an receptor binding sites for initiation of infection. This is quite important. So what happens if you can somehow destroy, by using some antiviral drugs, if you can destroy these surface structural proteins, then the virus will not be able to establish infection. So that might be an antiviral strategy of certain antiviral drugs. Okay. Similarly, as you say that if you use some alcohol-based hand sanitizer, it is very, very effective. Yes, this is quite true. If you think about this envelope viruses, this envelope is made up of phospholipids, and when they come in contact with alcohol, immediately this lipid get dissolved and all these spike proteins will remove all of the virus and virus will be inactivated. So the envelope viruses are more susceptible towards these detergents, towards the alcohol-based sanitizer in comparison to the adenoviruses. This is quite stable into this effect. Okay. So the structural protein basically protects the internal genomes of the virus. Okay. Not only that, some of the structural protein assist to migrate the genome from the cytoplasm to the nucleus for initiate its replication. So they also helps, the structural protein also helps to correct compartmentalization of this virus nucleic acids for genome replication. Similarly, the function of non-structural protein, if you see, they are mostly associated with the genomic replication, gene regulation. Some of the viruses, it is so interesting that they produce a single protein from their entire genome length. So after producing a single polyprotein, some of the enzymes present in the polyprotein will cleave it at respective sites. That will give rise to different structural and non-structural proteins. We'll discuss at the later part about this point. So these are the functions that you can see. It can modulate the whole innate response, assisting assembly. These are the work of functions of non-structural protein. Okay. So the viral proteins, we can categorize them as a structural and non-structural protein. Going to the third chemical compositions or components of viruses, you'll find lipid and carbohydrates. So basically, this lipids and carbohydrates you will find among the enveloped viruses. Rarely you will get in the non-enveloped viruses the lipids and carbohydrates. The lipids is basically it's the phospholipids, and that is the envelope layer, and they simply acquired it from the host cell during replication. This is quite interesting. In the later part also I'll mention about this aspect. You'll find there are certain viruses which grow and multiply within the host cell, and finally they comes out from the cytoplasmic membrane. And those envelope viruses will simply carry the cytoplasmic membrane of the host cell. However, at the same time, there are certain viruses which will come out from the Golgi apparatus or from the nuclear membrane. They will come out. As a result, they will acquire the nuclear membrane or Golgi membrane. So depending on the site from where the virus will come out, the envelope viruses, the envelope material will be of that nature. Okay. So obviously, this the glycoproteins, some carbohydrate molecules get anchored in the peplomers, and as a result, you'll find those carbohydrates, and basically 20 to 35% of the dry weight of envelope viruses you'll find is the lipids in nature. So these are the different chemical composition of viruses. Okay. Most simple, don't have much complications. So they had a very simple life, I should say, but the level of devastation, you can see what they can cause. So when we talk about the morphology and the structure of viruses, then you'll find these capsids, they are formed by certain monomeric proteins called as capsomers. Capsomers are the individual capsid proteins which will give rise to the entire layer, capsid layer. So the arrangement of the capsomer to form the capsid layer give rise to a definite shape to the virus. We call it as a symmetry, viral symmetry. Okay, you can note it down. What do you mean by viral symmetry? The arrangement of capsomer to form the capsid layer gives us a definite shape to the virus. We call it as a virus symmetry. So all the viruses we can categorize into basically this two symmetry, icosahedral symmetry and an helical symmetry. This is a certain geometric pattern they follow to assemble during their replication inside the host cells, and depending on their nature, the genomic coding proteins, the virus, they may appear as icosahedral symmetry or helical symmetry. So when anybody asks you about the shape of the viruses, you can say the viruses belonging to either icosahedral or helical symmetry. However, there is a category of viruses, what we call the pox viruses, who do not follow either icosahedral or helical. There is no symmetry. They simply aggregates and what we call as the complex type of symmetry. Of course, complex is not a symmetry. So basically, icosahedral and helical symmetry. So when we talk about the icosahedral symmetry, you'll be wondering about this, what is an icosahedrons or icosahedral symmetry? This is quite interesting, like even the physics people, the scientists from that lines and architecture and physics, they says that the icosahedrons are the most rigid structure in the world. That is why the nuclear reactor, inside the nuclear reactor, they designed it as icosahedron, because it can withstand a heavy pressure and energy. Now, how the viruses came to know about it, that nobody knows. But most of the viruses, they assembled just like an icosahedron. Now, when we call it icosahedron, it is a geometric configurations, which is having your geometric configurations, which is having 20 faces of an equilateral triangle. They're having 30 edge and 12 vertices. So when we talk about an icosahedron, we say that an icosahedron is a geometric structures composing 12 vertices. Vertices means this green color dot, you can see this is called vertices. These are the vertices. So an icosahedron is basically having 12 vertices, 12 in total, 12 vertices you'll find. It's a three-dimensional structure. Then 20 faces of an equilateral triangle. These are the equilateral triangle. I mean, all the sides are of the same measurement. Equilateral triangle, 20 faces and 30 edges. These are the edges of the icosahedron. So this is the definition of an icosahedron. Icosahedron is a geometric structure which is composed of 12 vertices, 20 faces of an equilateral triangles, and 30 edges. So this is the basic icosahedral symmetry. So the icosahedron viruses, they assemble in this particular fashion. So here you can see on the icosahedrons. So the proteins that presence in the vertices, as I say, the 12 vertices are present in the icosahedrons are called as penton protein, P-E-N-T-O-N, penton protein, because these are surrounded with 5 different proteins. Suppose the protein present in this vertices, it is taken it from the top view, this particular area. You'll find there are 5 different protein, adjacent protein. They surrounds 5 proteins surrounds this protein present in the vertices. So this protein is called as the penton protein, whereas all other proteins that forms this triangulation of the capsid layer are called as hexons, because you'll find they are surrounded with 6 different proteins. So the capsid proteins, we can describe them as the pentons and hexons. So pentons are the proteins that are present in the vertices, and rest all other capsid protein are termed as hexons. Okay. Now this is another diagram showing the 3, 5, and 2-fold rotational symmetry. If you can imagine on line or axis through the center of an edge. This is the edge, these are the edges. Any edges at the center of the edges, if you imagine, you'll get 2-fold symmetry. This is one, and the reverse sides you will get the other. This is called as a 2-fold symmetry. Whereas in the pentons, if you take the imaginary axis, then you will get 5 different rotational symmetry. And at the center of the triangulation phase, if you consider the axis, then you will get 3 different fold of symmetry. This is what we call as the 2, 3, 5-fold rotational symmetry. Now again, when you talk about the each triangular faces, the simplest icosahedrons where they are having only one triangular faces, we call it as a T1. However, it has been seen in certain other icosahedral symmetry viruses, each triangular faces are again divided into certain facets. The number of facets, some foldings are there in these triangular faces. So depending on the number of facets, it is described as a triangulation number, or this is an example of T4. Triangulation number 4 means 4 different facets are present within one triangular face. So these are helpful in sometimes calculating the number of capsomer presence in the icosahedral symmetry. Okay, some examples are given here with some simple formula like 10t plus 2 is the number of capsomer presence in that icosahedron. Okay, just for information I'm giving it. Now coming to the this two, the one is this icosahedral symmetry and this is the helical symmetry. In the helical symmetry, what the arrangement is just like a cylinder, this individual capsomer, these are the capsomer. This capsomer are arranged in such a way that they will give you a cylinder form, cylindrical form. And inside the cylinder, in the hollow cylinder, the nucleic acid remain protected. So this is called the helical symmetry. So most of the viruses, either they're icosahedral or helical symmetry, except the pox, which doesn't follow any kind of symmetry. So this is another picture showing penton and hexane proteins. In the pentons, like in the icosahedron, they are surrounded with 5 different proteins. That's why they call as the penton proteins. Whereas other proteins, you'll find 6 different proteins are surrounding each other and are called as the hexanes. Okay. So this helical symmetry, in case of the envelope viruses, the additional matrix, this envelope layer, and just below the envelope layer, we can get a matrix protein layer that gives us additional rigidity to the virus. Remember, this this envelope is similar to that of the membrane from where the viruses comes out. So obviously, the biological membrane, which is a phospholipid bilayer, exactly the same nature you'll find in the envelope viruses. So this envelope material is not of viral origin. The viruses doesn't have any genetic information for synthesis of these phospholipid bilayers. They simply acquire it from the host cell during its replication. However, certain glycoproteins or spike proteins like hemagglutinin, neuraminidase, or other spike proteins, these are of virus origin. They remain anchored, just like stabbing. They remain anchored into the envelope and they are projected outside the surface, as we could see in the envelope viruses. So these are the most basic structures of virus. Beside this, as I said, they don't have any cytoplasmic soup or any organelle, nothing. It is simply the nucleic acid structures. In case of the envelope, they're having the additional envelope layer. So these are some of the arrangement, how it is showing that they form the cylindrical form. This is called as the helical symmetry. So this SARS-2 COVID virus is an helical symmetry virus. This is the influenza virus, and this is a paramic Joe virus, where the nucleocapsid structure, it is helical. You could see some helical fashion, okay, just like a cylinder, the nucleocapsids form. Yeah, these are the different shapes of the category of viruses: double-stranded DNA, envelope virus, herpes, African swine fever, S4 viridi, then HIPAA DNA, and pox. It's a complex symmetry, doesn't have any definite symmetry. However, these all are icosahedral symmetry: papilloma, adeno, purple, circo. So these are different shape of viruses under the RNA category. So you can refer to any books where you'll get these pictures. And yeah, thank you very much. This much I prepared it for today's class. in the subsequent class, we'll go for, maybe tomorrow we are having another session where we'll go for the replication of viruses. Okay. So do you have any questions, specific questions related to this class or maybe the previous class? Can have some sort of interaction. Lema Thoi, do you have any question? Prem Agarwal. Hope that you're not going in the sleep mode. Brukesh. You can switch on your microphone and can talk with me. Okay, if you don't have any specific questions, then I'm just closing the link for today. Okay, bye-bye. Take care.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.